Okay, yeah, so thank you so much. So I am following up that wonderful talk with like slight different angle. So recently in our lab, we have been doing more white matter structural and microstructural imaging. So I'm going to try to, in this very short time, kind of relate those to actual vascular physiology. So it is well established that white matter atrophy takes place in normal aging. And so gray matter atrophy is well documented as well, and it's actually surpassed by that of the white matter. And in fact, the white matter atrophy is more closely related to cognitive decline in normal aging. So as you can see here. So white matter is usually not looked at in these types of studies for obvious reasons. I mean, physiologically, it is very poorly perfused. So what happens if you look closer at this atrophy? So microscopically, so microscopy was performed on these rats, younger and older, to demonstrate this microstructural change. And there seems to be an enlargement of the axons, and the black represents the myelin sheath surrounding the axons, and there's also been report of increase in average fiber length. And so isn't that sort of paradoxical? Why would anything increase, right, morphologically? So the question that we have is how do these compare? And this question has indeed been looked at before. So microstructure is commonly assessed using diffusion tensor imaging or DTI. So the chief parameters that are usually cited are mean diffusivity, MD, or fractional anisotropy, FA. So a lower FA and a higher MD have come to represent microstructural degeneration. And in a previous study by Light and colleagues, white matter microstructure in middle age has been found to predict later morphometric loss, so atrophy. So one question that we have is, in a cross-sectional study, for example, would we see in structures that survive that atrophy, so in the sort of tracts that remain, would we see that the tracts that sustain more volume loss also demonstrate more advanced microstructural decline? So that's the first question. That was a natural question for me. If we look yet closer, so what is feeding this degeneration, right? So this is a slide I just stole off of my good colleague, John Polimeni. So on the left, you can see sort of the typical tractography figures where you have the projection fibers, association fibers, and the commissural fibers here represented by largely different colors. And coincidentally, on the right, using this microradiographic depiction, you can see there are these medullary arteries that largely follow the orientations of the fibers. They're very much everywhere in the white matter. And the perforating arteries. So together, these two types of vessels feed the white matter. And as I mentioned before, it is notoriously poorly perfused. And so the question is, how does aging affect these arteries or these blood vessels, and how does that actually relate to microstructural changes, which we knew before is likely to precede the actual morphometric changes in aging? So to put that together, so years, actually decades of research without using imaging, in fact, has provided evidence for this sort of theory that we kind of put together. So in aging, we know that there's sclerosis in the vasculature. There's also loss of smooth muscle cells, as well as loss of astrocytes, and these are all taking place of negatively impacting the neurovascular unit. I mean, this is not an exhaustive list, but this type of vascular change is known to induce regional hypoxia, right? So in the previous talk, saw that it is possible with reduced blood flow, there is compensatory mechanism. But on the other hand, it is also very likely that the cells might simply starve of oxygen. So what happens when they're hypoxic in the white matter? On the one hand, there's evidence that the oligodendrocyte progenitor cells that differentiate into full-fledged oligodendrocytes, which constitute the myelin. So this sort of differentiation is actually prevented by the lack of oxygen. At the same time, or consequentially, there's also failure to repair damaged myelin so there's no incoming new myelin. So basically over time, there's demyelination, therefore microstructural degeneration. So this is our working theory. And the question is then, within imaging of the in vivo brain, do regions with lower perfusion also exhibit reduced microstructural integrity? Right. So perfusion has this, up to very, very recently been difficult to measure in the white matter. And so even now, we're still not sure, the integrity of the perfusion measurements compared to in the gray matter, but the Human Connectome Aging Project has really given us a possible window to look at addressing this linkage between morphometry, microstructure, and perfusion. So we have, through very strict screening for image quality, we ended up with 542 subjects, roughly an even split between males and females, and their age span is 36 to largely 90, with a few stragglers between 90 and 100 years of age. So our diffusion sequence is 98 directions with 2 shells, 15 and 1500 and 3000 seconds per millimeter squared. And so we use free-surface tracula to kind of as our working tool to generate DTI metrics, and also to provide us with subject-specific tract segmentation. So that's right. We're focusing on analyzing these not by white matter regions, but the actual known tracts. And so how tracula works very briefly. So within each subject, these streamlines are generated and then they're pruned to give us this native space set of segmentations for these well-known tracts. So I know with the recent release, there's an increasing number of tracts that are available. But for our analysis, we'll focus on these 18 main tracts, including these projection fibers, including the superior longitudinal fasciculus, which goes, there are 2 branches, the parietal branch and the temporal branch. There's the corpus callosum, largely represented by the minor forceps and the major forceps seen in blue and pink here. I'll come back to this later. So this just gives you a taste of sort of our spatial segmentation. And so with these, we first looked at the morphometry of the tracts, right? So we saw overwhelmingly there's a difference between sexes, and this is collapsing across all ages. And we see there are significant differences with men having higher tract length as well as tract volume, as indicated by the asterisks. So knowing this, we then looked at the tract, so normalizing these tract volume and length. So the way that we did this was we chose a volume reference here. In this case, for volume metrics, the conventional way is to actually normalize the intracranial volume, which are obviously different between men and women. And for tract lengths, we normalize by one selective structure, so the length of which does not really vary with age. So sort of this is how we selected the normalization reference. And to come back to this, then the normalized length and the normalized volume remain different between sexes, right? So they still all vary. So this is to be taken as sort of, for example, how large is someone's corpus callosum to the rest of the brain? So this is also different. The proportion of each tract relative to the brain size also differs between men and women. So then coming to the microstructure now, so you can see mean diffusivity on the left and fractional anisotropy on the right. So there is much less difference between sexes. And so this is to be expected. So these are largely similar between males and females. So then we have another way of normalizing our sort of to really target the mechanism of degeneration. We have these 2 different ways of normalization. So one, as I mentioned before, and the other one is actually based on referencing to the youngest group in this population. I will come back to that in a little while. So if we normalize by the anatomy, so in terms of proportionately how much a structure is losing length in the aging process, we can see So for the majority of tracts, there's significant loss of length in aging, whether we look at it as a raw length or as a proportion of the stable brain structure. So this tract is not only getting shorter, it's getting shorter relative to all other tracts. That's sort of what this represents. And so we'll see that the cingulum bundle in this case is actually most preserved in aging lengthwise, whereas the minor forceps and the uncinate fasciculus and cingulum angular bundle, which represents more the posterior cingulate, lose most length. So this is sort of one way to look at a tract by its length. Now, if we look at volume then, so length and volume don't necessarily coincide in aging, because a tract might well lose length but not get thinner. But the reverse is also possible. So if you look at tract volume decreasing with advancing age, the same is true. There's this great disparity across these different tracts. And so again, the cingulate seems to be most well preserved in terms of volume as well, but the major forceps exhibit the greatest volume loss. So major forceps are at the back of the brain. So this actually coincides with this well-known sort of theory about aging, is that there is this anterior to posterior brain gradient, wherein the posterior parts of the brain actually suffer more. The anterior part of the brain actually suffer more in terms of aging. So but this is sort of if the major forceps and minor forceps are both exhibiting volume loss, that they're at the two ends of the brain, right? So anterior and posterior both. So this is, an interesting new piece of evidence for us. So to actually approximate a tract as sort of a cylinder, we came up with this metric that is sort of the volume divided by the length. And so this is sort of looking at how much thinning the tract sustains in aging. So almost all tracts become thinner in aging, with the exception of the CST, which stands for this the corticospinal tract. So the corticospinal tract is among the first to develop in the brain, perhaps the first. And so it is also well known that is relatively preserved in aging. So far we've looked at the morphometry, and so I should mention that this work is all done by Tyler Robinson in my lab, who is completing his, who is in the middle of his postdoctoral fellowship. And so this is being prepared as we speak into his manuscript. So the corticospinal tract is the most preserved, whereas the minor forceps lose the most thickness. So the minor forceps, which are in the front of the brain, they thin the most. So as I mentioned before, there are two ways in which we normalize these metrics in view of the sex differences. And so the second way is to actually reference everything to the youngest group. And so we have done this for the morphometry, for the microstructure, and we'll do this same sort of normalization later for the perfusion. And so basically we use this to approximate a % loss in all of these parameters, using the youngest group as sort of the pseudo baseline. And so if you look at things this way, where I have listed the length loss effect size in terms of amount of loss per year, and the volume loss. And so both of these are quantified as percentages, and you can sort of start to see some trends emerge. In some cases, some tracts lose length and volume, meaning that they get shorter and they thin out. And these are mainly the minor forceps, the cingulate angular bundle, and the uncinate. So the minor forceps are in the front of the brain. So that's to indicate sort of this anterior to posterior gradient of aging, in which the anterior suffers more, as I mentioned before. But the CAB and the uncinate, as I indicate with this arrow here, they're not anterior, they're more temporal. So the temporal lobe, what is its role? We can also see a different manner of sort of morphometric loss is in terms of tracts that thin but do not lose length as much as they thin. So examples of that include the major forceps, the anterior thalamic radiations, and then the anterior cingulum, as well as the temporal branch of the superior longitudinal fasciculus. So there are these two trends that emerge for us. But what's more interesting is the CST, the cortical spinal tract, is sort of a pillar of aging in the sense that it doesn't seem to be as affected as the other tracts. So in terms of microstructure, the same comparison is made where you have mean diffusivity increasing and then FA decreasing, both indicating degeneration. While these two do not increase or decrease at the same rates, these are again percentages, they do sort of roughly mirror each other in terms of structures demonstrating more diffusivity increase also demonstrate more FA decrease. So the main structures following this trend include the minor forceps, which are again in the front of the brain, the cingulate, anterior cingulate, and the inferior longitudinal fasciculus. And all of these actually tend to be more, as I show in the yellow arrows here. So the majority of these tracts are in the front of the brain. But we also see if we were to combine these two to directly compare morphometry with microstructure, then we see sort of the structures that demonstrate the most morphometric loss, as we indicated before, definitely do also demonstrate significant microstructural degeneration, as indicated by these DTI parameters, right? But there are also those that demonstrate less length loss, as we mentioned before, a different type of morphometric difference. So tracts that demonstrate mostly length loss, mostly volume loss. In other words, they thin more than they shorten. So we don't know what stage they are at in terms of the morphometric sort of shrinking, but they are also demonstrating significant microstructural changes. So microstructural degeneration effects are definitely, they tend to be more robust. So you might think that these two methods are inherently different in sensitivity, as methods usually are. So what does that imply? So of course, we take that into account. We should, when interpreting these results, right? So you cannot compare apples to oranges, but at the same time, it's not like one of these methods is uniformly more sensitive than the other. So there is variation. So within this variation, within each method, what you should note is that, so by and large, all structures on this side, so in terms of microstructure, do demonstrate significant age effects. And some of these are lower than the effects demonstrated on the left-hand side in terms of morphometry. So that being said, so we do see that no matter how the morphometry changes, even for the corticospinal tract, there is still significant microstructural degeneration. So this, of course, is preliminary. We're writing this up and also preparing for additional analysis. So to summarize for this part, so what's interesting to us definitely does not fully answer the question, do more microstructural changes precede morphometric changes? Is this something that's generalizable? We don't know, but in our data, we see evidence that there are no tracts that show morphometric but no microstructural changes. So basically, every tract shows microstructural degeneration in aging. So this goes towards supporting that microstructural aging does take place more consistently and is more widespread. So to go to the next step, we bring perfusion into the picture. So blood flow, cerebral blood flow, or CBF, this has been shown to be, well, this has been investigated, in fact, in healthy young adults as far back as 2011 from Heng Zhang's group. And the results at that time were, so tracts with higher blood flow also demonstrate lower MD and lower FA. Perhaps because the tracts are more tightly bundled and there's not as much space for the blood to travel. So is that what we saw? First question for us is to see how far we can reproduce this result. but we're not using the exact same arterial spin labeling technique. So we're using within the HCP aging project, multi-delay pCastle, so pseudo-continuous ASL, with these are being the multi-delay inversion times. So the same subjects, and then we not only had access to the blood flow, but we also had access to the arterial transit time. So arterial transit time indicates when the actual labeled blood bolus peaks in the tissue region of interest, right? So it's a surrogate to let us know how quickly the blood is flowing, not just how much blood is reaching that region. So to demonstrate this, to try to reproduce what Aslan and colleagues had shown before, in our youngest group, we demonstrated that when there's a high CBF, higher blood flow, there's a significantly lower mean diffusivity. So this coincides with what was reported before. We actually did not see a significant association between blood flow and FA in our case. But bear in mind, this blood flow is not the same as the blood flow you would measure using single delay ASL, right? So I'll talk more about that. So to that point, we also try to associate arterial transit time to microstructure, and we found higher arterial transit time, so more delayed blood flow actually corresponding to higher FA. So this could actually be consistent with the previous results from Aslan and colleagues, in the sense that tracts that are more tightly bundled may just have lower flow rate. So this is sort of potentially consistent. Now looking at blood flow as a surrogate for vascular density. So I showed the medullary arteries before. So what we're really interested in perhaps is not only blood flow, but also just how much blood there is in these different white matter tracts. And so I am comparing side by side here. On the left are our baseline blood flow results for our various white matter tracts. On the right side are sort of a similar set of results, but using ferromoxetol enhanced imaging to actually map out the vascular density. So this is work from John Polimani's lab, Michael Bernier, shown at ISRM. And there's not a direct similarity that I could see, aside from the corpus callosum, in this case, the minor forceps and major forceps having the highest flow, as well as a high, relatively high vascular density. So this comparison, to me, it's not a direct comparison, but of course they are not meant to be equivalent. But they are both different aspects and both valuable aspects of vascular health. So coming back to how we found blood flow to actually vary with age. So using the pseudo-continuous multi-delay pseudo-continuous ASL, we actually show this nonlinear trend that we have not seen before. So this is over an age range of 40 to 90. And so initially blood flow seems to be stable, and then it just sharply declines over age. I must say that there had been suggestions from my earlier paper showing that after the age of 60 or 65, there's a much more drastic decline, but there wasn't really this sort of stable period in the early or to middle age that was reported before. One must wonder if this is because of the multi-delay versus single delay. But so there are differences across different regions in terms of how much the perfusion goes down. So the biggest loser in this case is the cingulum angular bundle, which encompasses posterior cingulum and then the tail towards the temporal lobe. But the major forceps, which are in the posterior part of the brain, also loses a lot of flow according to this. So now turning to arterial transit time. So as expected, so this transit time increases with age. So blood flow seems to slow down. And you can see the main structures affected, these two, forceps minor and ATR, are both in the front of the brain. So this is more in line with the anterior posterior theory of brain aging. If we were to put these together next to each other, we can see, again, some interesting trends start to emerge. Certain tracts show CBF reduction without a delay lengthening or ATT lengthening. So for example, these. And other tracts show the opposite. So the red tracts that show more blood flow decline without delay, perhaps are the ones that truly are suffering from hyperperfusion. So these include the major forceps, posterior cingulate, which CAB, and the inferior longitudinal fasciculus. And if you recall, these are similar to structures that I showed before, also demonstrating microstructural decline. But I will come back to that as well. On the other hand, there's an opposite trend. So structures that show more delay in perfusion, but without an actual significant reduction in diffusion. So the blood is coming, it's just late. So these structures include the minor forceps, the anterior thalamic radiations, and the anterior cingulum. So what does that mean? Again, so this is preliminary, but I would like to sort of use this data to raise some questions for you all to think about. Perhaps the fact that the red boxes show both microstructural and CBF declines in aging. So these are major forceps, the cingulum angular bundle, which is more the posterior temporal branch of the cingulum, and also the inferior longitudinal fasciculus. The opposite trend is tracts that show both microstructural degeneration and ATT lengthening. They include the forceps minor, the anterior cingulate, and tracts that are relatively spared in terms of microstructure. This was the cerebral spinal tract, but it does still show significant blood flow reduction in terms of both CBF and ATT, these two dimensions of perfusion. Right, so these are trends that we observed, and so what do we take away from this? This is still work that's ongoing. Hopefully will be presented at ISRM next year. But so to begin with, we started here on the right-hand side with morphometric aging. So tracts that morphometrically lose volume come in three flavors. They could shorten and thin, or they could shorten only or thin only. So we saw primarily, right, the first and the third categories. So what does that mean in terms of its linkage to microstructure? When we put the microstructural aging patterns against the morphometric aging patterns, we show that tracts that show morphometric but not microstructural age effects do not exist. Right, so this is potential evidence that microstructural degeneration may precede morphometric age effects. Finally, with the preliminary perfusion results, we show that tracts that show microstructure but no perfusion age effects also do not exist. So perfusion is affected in all tracts, even those that do not show or show a very moderate or mild microstructural decline. Right, so we take this as potential evidence that perfusion changes may precede microstructural changes. And what we're doing now is using decade-wise shifted analysis to actually further bolster the interpretation of these trends. So I wanted to just thank Tyler, who sort of is the lead on this project, as well as Paul, who has contributed a lot to the analysis. There are two members of the Cranium Lab, and also Jordan and Lydia, who have been involved, as well as our main collaborator, David from the MGH, and my funding sources. So thank you so much, and I'm open to questions.